Nucleic acids. Nucleic acids were first isolated in 1869 by Friedrich Becher from the nuclei of pus cells. Due to their isolation from nuclei and their acidic nature, they were named nucleic acids. Nucleic acids are of two types: deoxyribonucleic acid or DNA and ribonucleic acid or RNA. DNA occurs in chromosomes, in the nuclei of the cells, and in much lesser amounts in mitochondria and chloroplasts. RNA is present in nucleolus, in ribosomes, in the cytosol, and in smaller amounts in other parts of the cell. Nucleic acids are complex substances. They are polymers of the units called nucleotides. DNA is made of deoxyribonucleotides, while RNA is composed of ribonucleotides. Each nucleotide is made of 3 subunits: a 5-carbon monosaccharide, a pentose sugar, a nitrogen-containing base, a phosphoric acid. Pentose sugar in ribonucleotide is ribose, while in deoxyribonucleotide it's deoxyribose. Nitrogenous bases are of two types: single-ringed pyrimidines and double-ringed purines. Pyrimidines are cytosine, thymine, and uracil. Purines are adenine and guanine. Phosphoric acid has the ability to develop ester linkage with hydroxyl group of pentose sugar. In a typical nucleotide, the nitrogenous base is attached to position 1 of pentose sugar, while phosphoric acid is attached to carbon at position 5 of pentose sugar. The compound formed by combination of a base and a pentose sugar is called nucleoside. A nucleoside and phosphoric acid combine to form nucleotide. Each nucleotide of RNA contains ribose sugar, whereas sugar in each nucleotide of DNA is deoxyribose. ATP is also an important nucleotide, used as energy currency by the cell. DNA or deoxyribonucleic acid. DNA is hereditary material. It controls the properties and potential activities of the cell. It is made up of 4 kinds of nucleotides, namely D-adenosine monophosphate, D-guanosine monophosphate, D-cytidine monophosphate, and D-thymidine monophosphate. These nucleotides are united with one another through phosphodiester linkages in a specific sequence to form long chains known as polynucleotide chains. Two nucleotides join together to form dinucleotide, whereas three join together to form trinucleotide. Nicotinamide adenine dinucleotide, abbreviated as NAD, is an example of a dinucleotide. It's an important coenzyme in several oxidation reduction reactions in the cell. In 1951, Erwin Chargaff provided data about the ratio of different bases present in this molecule. This data suggested that adenine and thymine are equal in ratio, and so are guanine and cytosine. Morris Wilkins and Rosalind Franklin used the technique of X-ray diffraction to determine the structure of DNA. At the same time, James D. Watson and Francis Crick built the scale model of DNA. All the data thus obtained strongly suggested that DNA is made of two polynucleotide chains, or strands. The two strands are coiled round each other in the form of double helix. Coiling of two strands is opposite. They are coiled antiparallel to each other. The two chains are held together by weak bonds or hydrogen bonds. Adenine is always opposite to thymine, and guanine and cytosine are opposite to each other. There are two hydrogen bonds between adenine and thymine pair, and three hydrogen bonds between guanine and cytosine pair. The two strands are bound around each other so that there are 10 base pairs in each turn of about 34 angstrom units. The amount of DNA is fixed for a particular species, as it depends upon the number of chromosomes. The amount of DNA in germ cells, that is, sperms or ova, is one half to that of somatic cells. All the information for structure and functioning of the cell is stored in DNA. For example, in chromosomes of bacterium E. coli, each of the paired strands of DNA contains about 5 million bases arranged in a particular linear order. The information in those bases is divided into units of several hundred bases each. Each unit is a gene, a unit of biological inheritance. The E. coli genome consists of more than 4 million base pairs, which code for at least 4,288 proteins. Haemophilus influenzae is the first microbe to have genome completely sequenced, and this was published on July 28, 1995. RNA or ribonucleic acid. Like DNA, RNA is a polymer of ribonucleotides. The RNA molecule occurs as a single strand, which may be folded back to give double helical characteristics. The nitrogenous bases form the usual complementary pairs, like cytosine with the guanine and uracil with adenine. RNA is synthesized by DNA in a process known as transcription. Types of RNA. three main types of RNAs: messenger RNA, transfer RNA, and ribosomal RNA are recognized. All these three types of RNAs are synthesized from DNA in the nucleus and then are moved out in the cytoplasm to perform their specific functions. Messenger RNA. As the name indicates, it takes the genetic material from the nucleus to the ribosomes in the cytoplasm to form particular proteins. Messenger RNA carries the genetic information from DNA to ribosomes, where amino acids are arranged according to the information in messenger RNA to form specific protein molecule. This type of RNA consists of a single strand of variable length. Its length depends upon the size of the gene as well as the protein for which it is taking the message. For example, for a protein molecule of 1000 amino acids, messenger RNA will have a length of 3000 nucleotides. Messenger RNA is about 3 to 4% of the total RNA in the cell. Transfer RNA. It comprises about 10 to 20% of cellular RNA. Transfer RNA molecules are small, each with a chain length of 75 to 90 nucleotides. It transfers amino acid molecules to the site where peptide chains are being synthesized. There is one specific transfer RNA for each amino acid. So the cell will have at least 20 kinds of transfer RNA molecules. Transfer RNA picks up amino acids and transfers them to ribosomes, where they are linked to each other to form proteins. Ribosomal RNA. It is the major portion of RNA in the cell and may be up to 80% of the total RNA. It's strongly associated with the ribosomal proteins, where 40 to 50% of it is present. It acts as the machinery for the synthesis of proteins. On the surface of ribosome, the messenger RNA and transfer RNA molecules interact to translate the information from genes into specific protein.